It's a real pleasure to be here. Thank you very much for the invite. I'm going to be talking today about using genetics-based causal inference methods, a particular method called instrumental variables for causal inference in public health and drug development. So when I say gene-based causal inference, I mean Mendelian randomization. Here's a quick disclaimer. I am a part-time employee of Novo Nordisk, but I'm here very much in my academic capacity, and everything I'm going to be talking about today is about published research in the public domain. So just to make that clear. Okay, so a quick summary of the talk. I'm going to introduce what Mendelian randomization is and what causal inference is from my perspective, and then discuss the sort of revolution that's taken place in MR, really because of the explosion in the availability of publicly available genome-wide summary statistics. Then I'm going to talk about the problem of pleiotropy, which is a sort of violation of the key assumption you need to do causal inference, and some of the pleiotropy robust solutions that have been proposed by myself and others. And then I'll touch a bit on how MR is done specifically in a drug development context. This is a high-level overview, a whistle-stop tour, if you like, of lots of different topics. So apologies if some people get lost along the way. It's hard to know how to pitch this talk, but I've given it my best shot. Okay, so I usually start off with this example when I'm teaching my students about causality. So I imagine that you were asked by a GP, a sort of local area doctor, to help analyse some data on patients in her register. So she has data on 1,000 patients, and she's measured their body mass index and their systolic blood pressure. And the doctor's interested in understanding the relationship between BMI and SBP. Specifically, she wants to know, does BMI predict SBP? And the second question is, if I treat my patients to lose weight, will this lower their blood pressure? So you plot the data and you find that there's a strong linear relationship between increasing BMI and increasing blood pressure. So does BMI predict systolic blood pressure? Absolutely. And that means that we can use someone's BMI as a tool to screen for potential high blood pressure. So we could ask them in an online survey what their BMI was, and then we could invite maybe people with a BMI over a certain level to come forward to have their blood pressure tested, which is a bit more involved. The second question, if I treat my patients to lose weight, then will this lower their blood pressure? And this question is a lot harder, and the answer is not necessarily, because it's a causal question. Now, here's a sort of funny meme about the difference between correlation and causation. But the key thing with this data is that it's sort of survey data. And because we didn't control BMI or systolic blood pressure, we merely observe them together in individuals. It could be that there's an unmeasured common cause of both BMI and systolic blood pressure that's the reason why we see a strong association. Here's a causal diagram that I'm going to use over and over again in this talk. I hope that it's fairly interpretable. So U represents an unmeasured confounder. Now, even if there was a causal relationship between BMI and systolic blood pressure, as represented by the direct causal arrow, then if we measured the association between BMI and systolic blood pressure, what we would get would be the combination of the causal effect plus additional bias due to confounding that we didn't control for. And what I usually do with my students now is I ask them what the man in the truck could do to work out he wasn't having any effect on pushing the truck. And after a while, we get to the point where some of them say, maybe he could stop pushing and see what happened. And the students come around to the idea that what you actually have to do is the man would have to run an experiment. He'd have to change something about his behavior and observe the result, and then look at the difference between how fast the truck was going when he was pushing and when he wasn't pushing. And that's the sort of essence of causal inference. Now, the sort of gold standard method of inferring causality in the sort of healthcare world is a randomized controlled trial. And in randomized trials, what we do is we allocate patients, in this case, for example, to receive a treatment or a placebo. And because we randomly allocate treatment and placebo to the trial population, this means that the treatment arms are going to be balanced with respect, approximately balanced, as long as the trial is sufficiently large, with respect to all sort of physical characteristics that those individuals may have. And that means that if we observe a difference in the outcome of those individuals across the treatment and placebo arms, then we can be almost certain that it's due to the treatment, because that's the only difference there is between those two groups. One took the treatment and one didn't. Yeah, I always like to think of Sherlock Holmes in this particular example here. You know, what one of his famous sayings is, when you've eliminated all that which is impossible, then whatever remains, however improbable, must be the truth. And that single thing that remains is the treatment. Just extending the causal diagram to the case of a randomized trial, here, hopefully you can see R represents randomization now. And suppose we were going to randomize people to take a BMI lowering drug, and then we observe their systolic blood pressure a year, 68 weeks later. Because randomization is the sole predictor of whether somebody receives the drug or doesn't receive the drug, then we've sort of killed that arrow going from any possible confounder U into the decision to take the drug. And that means we don't have any confounding to deal with, and we can interpret the outcome, the difference in outcome between randomized groups as the causal effect of taking the treatment. Now, it's not always possible to do a randomized trial for various reasons, ethical reasons, cost reasons, and so often we have to sort of fall back on so-called pseudo-randomized trial evidence, and that's where genetics comes in. And what we typically do is we try to use someone's genetic data as an instrumental variable. Here, in this causal diagram here, G is now our genetic instrument, and it satisfies three important properties, or IV assumptions. The first is that it's robustly associated with the exposure X. The second is that it's independent of any confounders of the exposure outcome relationship. And the third assumption is that it only affects the outcome through the exposure. Now, note in this causal diagram now that G, unlike randomization, G isn't the sole predictor of the exposure X. There's an arrow going from U to X, but what we hope is that it's an independent predictor. So it gives us an independent prediction of the exposure. Now, to find genetic predictors for BMI and any other trait really that we can measure, we do what's called a genome-wide association study. And a genome-wide association study is done on a large number of people, say in UK Biobank, a population cohort based in the UK, which recruited 500,000 people in around 2010. We can take a sample of blood and then use a gene chip to measure their genome at about 1 million loci across all of their chromosomes. And then we can look to see how strongly each one of those genetic variants is associated with BMI. And the genes, the regions that you're seeing lit up here are the ones where we see the strongest hits. And there are around probably more now, but certainly a couple of years ago there were at least 100 independent genetic loci that were so-called genome-wide significantly associated with BMI. Now, you must understand that genetics only explains still a small amount of variation in body mass index, about 5%. So 95% of it is still down to your lifestyle, etc. So a small amount is due to genetics. And BMI is no different to a host of other traits. Pretty much every human trait now has a genetic basis. So there have been thousands and thousands of genome-wide association studies published. and pretty much all the way along the human genome, you'll find genome-wide significant variants for a particular trait. And that means that you can do the type of analysis that I'm going to describe, not just on BMI, but on almost assessing if the potential causal effect of any trait on any other trait or any disease. But to go back to the BMI example, the way that we do Mendelian randomization or instrumental variable regression to estimate the causal effect of an exposure X on an outcome Y is so-called two-stage least squares. So what we do is we regress our exposure BMI on our set of 100 genes, and that gives us a genetically predicted version of the exposure, which I've called X hat in the causal diagram now. And then I regress the outcome on that genetically predicted exposure. And because those genes, the instrumental variable assumptions tell us that the proportion of someone's BMI or exposure that is genetically predicted is independent of anything else, then that means that relationship is unconfounded. So that's the sort of theory of the standard theory of instrumental variables. Now this approach actually requires individual level data. So it requires a data set where you have measured someone's genetics and the exposure you're interested in, like BMI, and the outcome that you're interested in as well. And it's not always the case that you have a single data set that has all of those things measured. So a much more sort of widely applicable version of this approach is called two-sample summary data MR. And this is an approach that can link different data sets that might have measured the association between a gene and an exposure, like BMI, and then a separate genome-wide association study that measured the association between your genetics and systolic blood pressure. So what you do is you piece together those two genome-wide association studies, first of all, to find powerful instruments for your exposure from an exposure GWAS, and then for the outcome GWAS, you measure those same chosen selected variance association with the outcome. So what you're doing is you're piecing together two separate cohorts where one you've measured the gene exposure association, one you've measured the gene outcome association. For each one of those genetic variants, you get a causal estimate. And then we use a technique called meta-analysis, where we simply calculate a sort of weighted average, an inverse variance weighted average of all of those individual variant-specific causal effects. And you can show mathematically that the estimates that you get from this method based on summary data are asymptotically equivalent to what you would get if you had the individual level data. Yeah, the nice thing about the summary data approach is that it's very easy to sort of understand visually, because what it really amounts to is looking at plotting the data on a scatter plot. So this is a scatter plot plotting the gene outcome associations, or in our case, gene systolic blood pressure associations, versus the gene exposure associations on the x-axis. So that's BMI in our case, and then fitting a linear regression with the constraint that regression goes through the origin. So there's a zero intercept, and that's a direct consequence of the IV assumptions. Yeah, so you can see that these data points have uncertainty attached to them. So it's like doing a linear regression with some accounting for the fact that those data points have some uncertainty attached to them. Now, where it gets interesting is that it may not be the case that all of the genetic variants that you're trying to use for your analysis are valid instruments. And one of the major problems in Mendelian randomization is the issue of genetic pleiotropy. And that's a scenario where the SNP you're trying to use as an instrument for the exposure actually has an effect on the outcome, or some proportion of its effect doesn't go through the exposure of interest. And that would violate one of the key assumptions for Mendelian randomization, for inferring, getting an unbiased estimate for the causal effect. Now, one of the first tests you can do to detect potential pleiotropy is look to see if there's more heterogeneity or more noise in the data than you might suspect. Then there should be there if all genetic variants were valid instruments. And you can tests for the presence of heterogeneity, which is likely to be caused by pleiotropy, with a heterogeneity or Q statistic. And if we see a large Q statistic, then that's when we can start to think about employing a pleiotropy robust method instead. So to account for the fact that your genetic variants, some of them may be pleiotropic, we need to extend our sort of causal outcome model so that it includes specific direct effects for the genetic variants. So in red there, I've shown, I've coded those as alpha j times gj. So the alpha term represents, if that coefficient is non-zero, then that represents that gene j has a non-zero pleiotropic effect. So that's an individual level data model. But when we calculate our summary data, what this means is that what we'll get is our SNP outcome association for our jth SNP is going to be follow this model instead. So it's going to have a potentially a pleiotropic effect, and then the causal effect multiplied by the gene exposure association. And if that alpha parameter is 0, that's okay. We've got a valid instrument. If it's non-zero, then it's invalid. And you'll note that this model is what's called under-identified. We've got more parameters than we have data points. If we have L SNPs, we've got L plus 1 parameters. So we can't estimate all of the pleiotropy parameters and the causal effect. So we've got to make another choice in order to infer what the causal effect is. We've got to assume something about the pleiotropic effects and the gene exposure associations in order to be able to identify the causal effect. Now, the sort of easiest thing that you can do is to assume that the pleiotropy is essentially random noise. So even if 100% of the variants were pleiotropic, but their pleiotropic effects were independent of how strong each one of those variants was as an instrument, and it was just adding sort of no bias but just noise, then you could calculate the standard estimate but simply inflate the standard error to account for the additional variability induced by the pleiotropy. And this would be called a sort of random effects model, a random effects meta-analysis. Now, another technique that was proposed in a paper by myself 10 years ago is to use a method called MR-EGR regression. And this is simply a sort of extension of the IVW approach, but where we include an intercept in the regression. And what the intercept should do is test for meaningful bias induced by pleiotropy in the IVW estimate. And the slope adjusted for a non-zero intercept is an unbiased estimate for the causal effect if those pleiotropic effects are independent of how strong those variants are collectively as instruments, even if the pleiotropy is inducing a sort of non-zero systematic bias in the data. Now, this method it gives you very imprecise estimates. So after it was proposed, lots of people realized this and thought about proposing other methods that could give you more precise estimates when applied to data. Okay, so the next robust method instead I'm going to talk about is the so-called median-based MR. And here's a quick diagram that tries to explain the rationale for using a median-based method. As we know, a sort of median, the median is a robust measure of central tendency. It's robust to outliers. And in this case, suppose we've got 13 variants, but 7 are valid and 6 are invalid, and we calculate the median causal estimate across those 13 variants, then that's going to be much closer to the truth than the sort of overall weighted average. I didn't show it, but this method is going to be consistent, if at least 50% of the variants are valid instruments. If we had infinite data, what we'd expect is those hollow dots there to line up along a line, and we would get a consistent estimate. But you can see even in this plot, which is sort of representing a sort of a finite data setting, the median will be more robust. There is an even more robust method, which is based on sort of mode-based estimation, and it works even if there's a very, very small subset of genetic variants that are valid. So here in this plot, we've got 8 variants, N0 to N7, and 2 of them are valid instruments, and the other 6 are invalid. But the 2 ones that are valid give you a very, very similar causal estimate. And it's possible if you, for example, try to characterize the density of ratio estimates using maybe a sort of kernel density approximation, that you can find the mode of that empirical density and then say that that's going to be your causal estimate. And so we proposed this in a paper shortly after, and it has this sort of very high degree of robustness. Even if a very small amount of your variants are valid, it will still give you the right answer. A related version of this is based on something called 2-stage hard thresholding with voting, and this is a method which sort of takes an analogy from optimal voting strategies in elections. And so what you imagine is that for each ratio estimate, you calculate its ratio estimate and its 95% confidence interval. And if you have a 95% confidence interval for variant A's causal effect, and it includes variant B, then that means that A, in effect, votes for B. And what you choose then is the largest group of variants that all vote for each other, and you select that as your sort of consistent subset of the data, and then you can calculate your standard IVW estimate on that identified sort of subset of consistent information. It's always, whenever I've thought about these pleiotropy robust methods, it's always made me think about Arrow's theorem. And since I'm at a mathematical institute, I think that this is a potential open question for anyone interested in proving something. So is there an Arrow's impossibility theorem for MR? Is there a perfect MR method? Or are all MR methods somehow doomed to fail some sort of criteria that according to this sort of theory of Arrow's on voting strategies? There's a very, very strong analogy that comes up again and again. So I just thought I'd mention that. Okay. Now, all the methods I've been talking about so far have mentioned trying to hone in on a sort of single causal effect that is measured in a data set, but often that is just too simplistic. And in the case of BMI, it's been known for a long time that BMI is a very crude measure of obesity, and it matters a lot where, if you are obese, where that body fat is. So you could have body fat in your extremities, around your legs, say, or you could have it around your central organs. And having central obesity is far worse for your health. And it's typically more men that have central obesity and women that have obesity in their extremities. And so it's absolutely the case that those 2 types of body fat in different locations in the body actually have different causal effects on downstream outcomes like systolic blood pressure. And if we were to only measure BMI, actually there's latent substructure in the data that we're not modeling. And what we would see potentially is a scatter plot that looked more like this, where we would see evidence of clustering in the data. And so, we would, in this case, we've got some red genetic variants that are actually coding for one obesity subtype that are identifying one causal effect, and then the black variants that are are predicting a different type of obesity, and they're identifying a different causal effect. And so what we do instead is try and use methods from cluster analysis to try and find out which, for a given variant, whether it is part of the red cluster or the black cluster, and then we simply calculate the causal estimates identified by both of those subsets. Here is one example of that we used I'll just skip to this slide here. we've taken on we've taken this method or this approach forward in several different directions, and we've proposed Bayesian methods to do soft clustering of the genetic data, and then also a method called agglomerative hierarchical clustering, which is a hard clustering method to try and delve into the data and find this latent substructure. The plots I'm showing here relate to the second publication in that list, the agglomerative hierarchical clustering approach, where we extended the idea also to multiple outcomes. So what we did is we tried to look at BMI, took around 500 variants in this case, and tried to find subsets of those genetic variants that had distinct effects on BMI and osteoarthritis together, so two outcomes. And we were able to do that successfully. So what you're seeing here is a sort of two-dimensional plot of causal effects identified by different variants, and then the color coding comes in using the agglomerative hierarchical clustering algorithm. Okay, so how am I doing for time? Am I okay? Yep, okay, good. So I'll move on now to talk about MR in drug development. I've been talking about BMI so far, and BMI is something that in public health we can measure and then try and propose strategies to help people lose weight, just in the community with maybe diet and exercise or other things. But in the pharmaceutical world, what we're trying to do is develop drugs to treat diseases, including obesity. And MR is used in drug development, and it's mainly used to try and identify proteins that are good drug targets. Now, yes, specific genes in the genome produce and regulate the level of different proteins in the body. And the main source of data that we use for this sort of analysis, again, comes from a subset of the UK Biobank data called the Pharma Proteomics Project. And this is a subset of 50,000 people in UK Biobank, where we have also measured not only their genetics, but the levels of circulating proteins for about 1,500 proteins from their blood. So we can link their genetics to around 1,500 proteins. We can try and use the principles of MR to find good drugs targets for a range of different diseases. Just to bring it to life a bit more, we can use MR to see if genetically predicted levels of a given protein are causally implicated in a specific disease. Using exactly the same principles. Now, whereas before, when I talked about instrumenting BMI, you would use that using so-called genome-wide instruments. You'd look across the entire genome and say, how strongly can I'll just use all the variants that are genome-wide significant for MR. But in this sort of drug target context, we use genes from a much more specific gene region. So if we're interested in a specific protein, there's a single gene region that will be the genetic code for that protein. And then we hone in on that single region and try to construct genetic instruments. Now, the statistical challenge from doing that is that genes in a tight gene region will be highly correlated due to a phenomenon called linkage disequilibrium. So they're much more likely to be inherited together due to the process of recombination that passes your genes from your parents to yourself. And so because of that high correlation, what we typically do is we employ statistical techniques like principal component analysis to try and create sort of principal component scores of all of the genetic data in a single region. That means we extract all of the data, and sort of encapsulate it within a set of principal components that are powerful instruments but are also independent. And because of that independence, then we've got back to our very easy to understand meta-analytic sort of structure of analyzing the data. The next thing we do when once we've looked at the link between a protein and a disease is we try to understand which tissue the gene is expressed in or produced in, because that might tell us specifically a kind of mode of action for the drug and where, how to tailor and target the drug even more specifically. And we can do that using tissue expression data to find the specific cell tissue type where that gene is most highly expressed. Now, this is another real statistical challenge when you're doing this sort of analysis, because often we want to try and simultaneously estimate the effect of expression of a single gene in multiple tissues, but we may have more tissue types than genetic instruments. And that means that we've got an under-identified model again. So here in this plot, we've got 3 genetic instruments and we've got 5 tissue types. And we can't fit that model if we want to estimate the effect of all tissue types, but actually this graph here is an example where only 2 of them have a causal effect, so only X4 and X5. So there is a submodel in the data, if we could find it, that is identifiable. And so, yeah, in recent research just published that's going to be presented at the AI Stats conference this year, we have proposed a sort of sparse approach to modelling this sort of data. And when we applied it to the specific example I talked about previously, we could identify 3 tissue types that were driving the expression of that gene and would be therefore good targets for further development of the drug. Okay, so thank you very much for listening to a whistle-stop tour of MR. I thought that I would also take a moment to acknowledge the Chinese collaborators that I've worked with over the last 10 years on some of the projects that I've described here. And also, to thank Huawei for inviting me and for sponsoring this event. Thank you very much.
Thank you for Professor Bowden's talk. Actually, I know a couple of them. Yeah. So, do we have any questions? Oh yes, please.
So I have 2 questions, and the first one is, I know that econometricians have used instrumental variables for a long time, and they are very worried about treatment effect heterogeneity, right? So they even say specifically that they estimate lates instead of average treatment effects. And I've noticed in the Mendelian randomization literature, this seems to not be something people worry about so much.
Yeah. Well, I mean, this example here is an example of when you're collectively an entire set of genetic variants you could think of as the union of set G1 and G2 would violate your standard so-called homogeneity assumptions. They're different instruments identified different causal effects here in this plot. And so, yeah, I absolutely agree with you that the MR has focused much more on detecting invalid instruments or pleiotropic instruments, and for many years just assumed that the homogeneity assumption was satisfied. But this sort of work is an attempt to try and uncover and model effect heterogeneity in a bit more detail.
I guess it's a different mathematical way of expressing it, saying there's 2 treatments rather than saying it's just a more complex, non-linear, non-additive function. But you see how they are probably equivalent. Yeah, I mean, there lots of methods have been proposed to try and model non-linear effects as well. Typically the genetic effects are very small, and they tell you about the effect of perturbing a trait like BMI, just a few 1 percentage point up or above or below the center of that distribution, and so there isn't that much information to learn about nonlinear effects. But no, that's a fair point. And then the other question I had is, because I've worked a little bit on conditional IDs, and one thing I was wondering, for example, is when you assume no pleiotropy and you have linkage disequilibrium, aren't you implicitly assuming that there's not just no pleiotropy for your individual instrument, but also for all the other ones in linkage disequilibrium with it? And doesn't that raise more questions about, should we adjust for these other instruments?
Yeah.
As a habit? In general, I've had a hard time understanding what do people generally adjust for and what don't they adjust for in these conditional IDs.
Yeah, you're right here. In this plot on the left here, plot A, we've got SNP2. It would be a valid instrument if you conditioned on SNP1, wouldn't it? So you can do that. You could calculate the conditional association between SNP2 and trait 3, conditional on SNP1, and that would be a way of blocking it's conditionally valid given SNP1, and that sort of approach is taken in MR analysis. Typically, what we try to do is take a single variant in a single gene region that's the top hit, and make sure that the variants aren't correlated with each other. But no, I absolutely agree that there could be a SNP could be in linkage disequilibrium with something close by, and there could be an effect. You usually follow up an MR analysis with much more in-depth genetic analyses, so-called co-localization analyses that try and really look in the genomic region either side of where you're picking your variant to try and understand if there are any other, if there are correlations that could lead to such sort of indirect pleiotropic pathways. Yeah.
So then the answer is people tend to not do it, but they are worried about it, but they tend to analyze it and follow up things, these co-localizations.
They tend to not do it, but then look at the potential impact. It's an underdeveloped area. Yeah, I think there's still better methods that could be proposed to try and find the best way of conditioning on variants in a region. Maybe not using a variant as an instrument, but using it as something you're conditioning on. We're not there yet with the perfect solution, absolutely.
Okay. Do we have any other questions? So if not, let's thanks to the Professor Bowden. Thanks for your...
All right, thank you very much.